So we've all heard about DNA replicating itself, and most of us probably don't even think about it often, but it's actually very complex and it points to design. So I talked to Dr. Jonathan McLatchie, who is a molecular biologist with a PhD in evolutionary biology. This episode is full of information that you're gonna have to pay attention to, but what he says at the end, I think that you're gonna really enjoy because I haven't stopped thinking about it. Dr. Jonathan McLatchie, thank you so much for joining us today.
Thanks for having me. It's great to be here.
One of the things that I love to ask is, of course, because so many people that are going to be listening to this are students, is how did you get interested in not only intelligent design, but biology as a whole?
Sure. So I did my undergrad degree in forensic biology at the University of Strathclyde in Glasgow, Scotland. And during my undergraduate years, I became very fascinated with the complexity and designoid characteristics of living organisms. I was blown away by the engineering prowess and elegance of cellular machines and was often bewildered at the fact that someone could go through a 4-year university program in the natural sciences and come out an atheist at the other end, because the evidence seems to be so compelling for the truth of theism, both in the life sciences, which is my expertise, as well as in the physical sciences as well. And then subsequently, I pursued a master's degree in evolutionary biology and another master's degree in medical and molecular biology. These were from the University of Glasgow and University of Newcastle, respectively. And then I did my PhD at the University of Newcastle in cell biology, with a focus on the eukaryotic cell division apparatus by which cells divide. Eukaryotic cells, that is, cells with a nucleus, like our own cells. So yeah, I've been fascinated for many years, in particular by the case for intelligent design, which I take to be very, very strong indeed.
With the topic of the science dilemma, we constantly want to point to how science points to intelligent design. And so that being one of our goals as a biologist, what's one big way that you think science points to intelligent design?
Sure. So we've discovered that the cell is chock full of digitally encoded information content that runs along the sugar phosphate backbone, or the spine of the DNA molecule, and this forms what we call the genetic code of life, where you have a language convention that in the language of DNA, you have triplets of nucleotides, or letters, triplets of nucleotides of these letters that form words that we refer to as codons. And there's 64 different codons, and these specify an amino acid, right? And there's a redundancy between there's more codons than there are amino acids, so there are many amino acids that are specified by more than one codon, and basically the sequential arrangement of these letters and these codons determines the sequential arrangement in turn of the amino acids that form the subunits of proteins. And then the way that these amino acids are arranged will determine how a protein will collapse into its three-dimensional structure, and that's determined by the properties of the side chains of those amino acids. So in every other realm of experience, when we find language conventions and information processing, storage, and retrieval apparatus, we habitually associate that with conscious activity or intelligent deliberative agency. And so when we find information rich structures, language conventions, information processing, storage, retrieval apparatus, and so forth, I think the best explanation is that it too arose by virtue of an intelligent cause, rather than an unguided process of natural selection and chance genetic variation.
Yeah, basically, anytime that we see, I mean, is that kind of similar to the concept of, like, if you put 1,000 monkeys in a room, they're not going to write Shakespeare? I think that we've heard that before, or something like that. Like,
yeah. So there's a large element of chance that is necessary for the evolutionary process to work. Of course, evolutionists will say that that evolution is not entirely a chance-based process, right? There's also natural selection, which is a non-random component, but chance genetic variations that we now know to be caused by mutations in the DNA are are sifted by this mill of natural selection. And natural selection is great at ensuring the survival of the fittest, but it's not so great at ensuring the arrival of the fittest. And so it turns out that there are many systems throughout life which exhibit a property that we call irreducible complexity. And that is to say, where you have various well-matched interacting parts to contribute to the overall function, wherein the removal of any one of those parts would essentially result in the system being non-functional. And so this is not only a challenge to evolutionary processes, because how would you, by a blind search, put together such a system one small step at a time when you require all the parts to be working together for the system to work. But it also points to design as being a better causal explanation, because only intelligent causes are able to visualize a complex end goal or complex higher level objective and bring together everything needed to realize that higher level objective, whereas unguided processes cannot. And so in view of that top heavy likelihood ratio, where these sorts of systems are not particularly surprising on the supposition of design, whereas they're wildly surprising on the falsity of design, we justify an inference to design. These are strong, in my opinion, cumulatively, it's overwhelming evidence for a design-based perspective.
Could you explain like what what way does irreducible complexity kind of flesh out within biology from your expertise?
Sure. So there are many such systems throughout biology, many thousands, in fact, and it's best illustrated by giving examples. So, I mean, I think you've had Behe on in the past, and he likes to give the example of the mousetrap, which is an example from everyday usage, where if you have the spring, and you have the hold down bar, and you have the platform, and you have the staples that hold the trap together, and so forth. If any of those parts were missing, then you don't have a trap that catches half as many mice as it used to, or a quarter as many mice as it used to, but it's broken. And so by an unguided search, without knowing what the target is, how would you put together such a system? By slight successive modifications. And so to take a biological example, one example is the DNA replication machinery. And the DNA replication machinery basically prepares the cell for cell division by replicating or duplicating the DNA molecule. DNA is the molecule that stores the cell's informational content. And that has to be passed on to successive generations. And so it has to be replicated or duplicated. And there's a number of well-matched interacting parts to contribute to that process. So, for example, you have the initiation proteins that open up the DNA double helix and prepare for copying. You have the helicase enzyme, which breaks the hydrogen bonds that link the two strands of DNA together, and that facilitates copying. You also you have to have what are known as single-stranded binding proteins, which bind to the single strands of DNA to prevent them from reannealing during copying. And you have to have the polymerase that actually performs the copying. The polymerase has to be clamped on to the strand to prevent it from falling off by a sliding clamp. And the sliding clamp has to be loaded by a clamp loader. And you have to have topoisomerase enzymes, because as the DNA gets replicated as a result of the torsional stress on the DNA molecule, this could result in supercoils. Think about an old telephone cord that forms these supercoils, and so that has to be alleviated. And there's a whole class of enzymes responsible for that called topoisomerases, which essentially function by cutting one strand, passing the other strand through the gap, and then resealing the break. And you have to have the primase enzyme, which synthesizes short RNA primer in order for the DNA polymerase to continue, because DNA polymerase is not able to begin copying without the presence of an available 3' hydroxyl chemical group. And so RNA polymerases don't require that in order to begin synthesizing. And a primase is a form of RNA polymerase, and so it synthesizes a short sequence of RNA called a primer. And you have to, because of the anti-parallel nature of DNA, with one strand, the strands being running in having opposite orientations or opposite polarity to one another, one of the strands, it's called the lagging strand, has to be looped around and copied backwards. That's how the replication fork moves in the same direction on both strands. So one of the strands has to be replicated backwards because the DNA polymerase copies in a 5' to 3' direction. Um, and for that to work, you have these short RNA primers that are synthesized on the lagging strand by an RNA primase, and then you have short fragments of DNA sequence called Okazaki fragments, named after their discoverer, that are synthesized from those RNA primers. And then the RNA primers have to get removed by RNA excision enzymes, replaced by DNA. The Okazaki fragments have to get stitched together by an enzyme called ligase, and so forth. And if any of those components are missing, then you don't have a DNA replication machinery that works half as well as it used to, or a quarter as well as it used to, but it's broken. You also, of course, have the telomerase enzyme, which because the DNA polymerase isn't able to finish copying all the way to the end of the chromosome, and so there's these repetitive sequences at the end of chromosomes known as telomeres, and it has a repetitive sequence of T, T, A, G, G, G over and over again. And the telomerase is a riboprotein that is able to synthesize these telomeres to stop the chromosome from shortening with each round of successive replication. And so again, if any of those parts were missing, you don't have a system that works half as well as it used to, or quarter as well as it used to, but it's broken. And it turns out that even getting individual proteins by mutation and selection is prohibitively difficult, especially in view of the rarity of protein sequences, stable and functional protein sequences within combinatorial space, right? It turns out that proteins are astronomically rare within the number of possible ways there are of arranging amino acids. And according to Doug Axe's estimate, for example, and it's not the only paper that makes this case in the literature, but his paper in 2004 in the Journal of Molecular Biology is a famous example where he argues that the ratio is in the order of 1 part in 10 raised to the 77th power. And these probabilities multiply exponentially when you require not just one protein, but you require multiple different proteins that have to work together in unison to bring about a higher level objective, right? The probabilities multiply together, and you don't need to see any old protein that will fold stably, but you need very specifically crafted proteins that have to work together in a very coordinated way. And given the primitive nature of DNA replication, because DNA replication is really necessary for differential survival, which is itself a requisite for natural selection. And so you can't really invoke the existence of natural selection to explain the existence of self-replicatability without assuming the existence of the very thing you're trying to explain. But moreover, because it's so primitive, unlike with some other examples of irreducible complexity, like the bacterial flagellar motor, for example, where someone could say, well, perhaps you could borrow parts from other systems that, such as the type 3 secretion apparatus, for example, that's like a needle that injects toxins into multicellular organisms and possesses maybe around 10 of the a few dozen proteins needed in the flagellar system. Unlike with a system like that, with DNA replication, what are you going to borrow from? Because it's so primitive, there's nothing that you can really borrow from. And so I think DNA replication is a really formidable example of an irreducibly complex system. And as I said before, this is not merely a challenge to evolutionary processes, but it also, I think, evinces design quite strongly as well.
And so if somebody ever was to try to say that, like combat where your stance is on using this for intelligent design, it's probably extremely difficult because you would need all of these things for irreducible complexity.
Yeah, exactly. And remarkably, it's been shown in the literature that the enzymes that are involved in DNA replication between the branch of evolution that leads to the eukaryotes and archaea and the branch of evolution leading to the bacteria, that the enzymes involved in DNA replication are not homologous to one another between those two lineages. And so it looks like DNA replication arose at least twice independently. And so that is, I think, exacerbates the problem even further. And I think points to design as being a much better and more elegant explanation.
Yeah, because the information already existed and it happened simultaneously. or just an
Right. I mean,
independent
it's enormously
moments?
unlikely that it could happen once, but happening
Yeah.
twice
Yeah.
with the same logic independently, that seems to strain credulity, unless in fact
Yeah.
there is a mind behind it.
Yeah. It's two miracles happening up that they weren't anticipating. So what does all of this mean when it comes to intelligent design? Like, what are we supposed to take away from this when we see DNA replication?
So, intelligent causes, as I said earlier, have the unique ability to visualize complex higher level objectives and bring together everything needed to realize those objectives. And so, on the supposition that a mind was involved, these sorts of systems are not particularly implausible. Notice that I don't have to say that these systems are highly probable on design. I just have to say that they're not highly implausible, whereas they are highly implausible on the falsity of the design hypothesis. And so, in view of that top heavy likelihood ratio, this is what we call a Bayesian inference. It favors a design perspective. And as you consider numerous examples like this of reducible complex systems and other features of life that evince design, like causal circularity, the rarity of protein folds, and various other things, it seems to point very forcefully to the hypothesis of design as being the best, most elegant, most probable explanation of the available evidence.
So, from understanding that about intelligent design and the implications that DNA replication has on that, I also wanted to pivot a little bit to the fact that you've not only done science, but you're a writer, you do debate, and you're also a professor. So, having with your experience in all of these different areas, why should the topic, and why do you usually tell people the topic of intelligent design matters for your everyday person like myself and other families that are listening?
Sure. So, intelligent design, I think, is part of a broader cumulative case for the existence of a creator behind life in the universe. So there's tremendous evidence of design not only in the life sciences but also in the physical sciences. There's evidence of that the universe seems to have had a beginning at some point in the finite past. That astrophysicists typically date to around 13.8 billion years ago, and there's tremendous evidence of fine tuning of the laws and constants of our universe for life to exist, in particular intelligent life to exist. And then there's also the argument from the prior environmental fitness of nature that Michael Denton and others have developed, which basically argues that there are features of the periodic table and properties of light and water and so forth that make life, and in particular advanced life, probable or possible rather. There's also, and then there's the evidence of design and biology, which is consistent with that. And so these sorts of features in the world are not hugely surprising if indeed a creator does exist behind life in the universe. Whereas they are wildly improbable if we suppose that there's no creator behind life in the universe. And of course, having a creator behind life in the universe is something which is consistent with what you would expect on the hypothesis of Christian theism. And I, of course, I'm a Christian, and I also happen to think that there's very, very formidable evidence for Christian theism, as well as just for general theism. But that would take us into another discussion. But if anyone among the listeners is struggling with doubts about the existence of a creator or the truth of Christianity, then I actually run a ministry separate from my work at Discovery Institute called talkaboutdoubts.com, where people can reach out and talk to a specialist, an expert, a PhD level scientist or scholar in a relevant field, such as in the natural sciences or in biblical studies, New Testament, Old Testament, biblical archaeology, textual criticism, philosophy, etc. And we do one-on-one private Zoom meetings to discuss people's doubts with them in confidence.
I love that, and I think that so many people would benefit from that. Even if they aren't Christians and they just want to learn more, they'd be able to reach out to you guys and just have the conversation.
Absolutely.
That's awesome. Well, thank you so much for diving into this topic with us and explaining how DNA replication points to an intelligent designer, and then what the implications of that would be from us from a worldview standpoint as well. Other than Talk About Doubts, is there anywhere else that people can engage with you?
Sure. I have a website at jonathanmclatchie.com, which houses a lot of my scientific writing, as well as my writing in other disciplines, particularly New Testament studies, which I've done a lot of work in. You can also find our our Discovery Institute blog at evolutionnews.org. I'm a frequent contributor there. And we also have a podcast, idthefuture.com, where I'm also a frequent contributor. Discovery.org/ID is kind of our homepage for the Center for Science and Culture, of which I'm a fellow at the Discovery Institute. And as far as books are concerned, I'd probably begin with Doug Axe's book, Undeniable: How Biology Confirms Our Intuition That Life is Designed. He writes very accessibly for a general reader. And Michael Behe also writes very accessibly for a general reader. I'd probably begin with his most recent book called Darwin Devolves.
Awesome. I feel like if anybody wanted to just talk to you about all the things that you do, we'd have years worth of podcast information.
Yeah.
I appreciate you so much, sir.
All right. Thanks so much for having me.
Have a good one.